All questions and feedback are welcome, and I've included my email here in case you wish to contact me later on. So let's start with an overview of this workshop. I will begin by sharing the aims of our work, and I will expand on these by giving you an introduction. And then I will introduce you to reduced order modeling, as well as some of the mathematics we used, and describe how we used it in our present study. After providing some background, I will explain the experimental methods used and do a walkthrough of the graphical results of data visualization and share a few conclusions as well. Then we will transition into a more interactive portion where you'll have the opportunity to share feedback and learn where to find and access the resources provided by PM3. I will conclude by just briefly discussing achievements and also future work. Although the Q&A period is at the end, I welcome you to type any questions that might come up into the chat, and we will just address them at the end. So the first aim of this work is to develop a reduced order rat model that permits the simulation of dialyzer-induced ischemia on the whole body and whole body hemodynamics specifically. Our goal is to develop a hemodynamic mathematical model of whole rat circulation coupled to a representative dialysis machine, and this can be used to inform future clinical data interpretations. And concepts relevant to this, such as reduced order modeling and hemodynamics, will be discussed further in the subsequent slides. So the second aim was to assess microvasculature network statistics over the course of small animal dialysis. As mentioned previously, Gabrielle Ali Morad led this work, and I encourage you to watch or revisit her presentation from yesterday, Saturday, August 14th, for a more in-depth overview of this topic. Globally, 2 in 7 people are living with chronic kidney disease, or CKD. And in Canada, more than half of all chronic kidney disease patients receive dialysis. This is because hemodialysis is one of only a few options for patients with end-stage renal disease. Renal disease is devastating and reduces the quality of life of the individuals afflicted with it due to the damaging effects of impaired kidney function. As a result, there is a large interest and investment in improving treatment options for patients suffering from this disease, including the optimization of dialyzer composition. So this diagram shows the primary functional unit of hemodialysis, which is the dialyzer, and it acts as what you might imagine as an artificial kidney. So this schematic shows the dialysis unit, and determining the optimal dialyzer composition can assist with improving patient outcomes because the dialyzer functions to remove uremic toxins and excessive water while retaining blood proteins. Currently, synthetic materials such as polysulfone are widely used. However, the selectively permeable nature of the polysulfone-based dialyzer membranes can elicit side effects. Organic polymers are regarded as safer alternatives for humans, but their use is currently limited because they prolong the duration of the dialysis session. So given these limitations, further research can assist with enhancing the design of these dialyzer materials and to help further reduce ischemic stress. Animal models, including rats, have been used extensively to study hemodynamic phenomena, and the rat serves as a suitable model of renal processes due to the similarities between the human and rat cardiovascular and renal systems. To construct the whole rat hemodynamic model, we are using a reduced order approach, and this method is commonly used for cardiovascular modeling and is known to be accurate for most cardiovascular simulations. We are building a 0D model which approximates the artery systems as electrical circuits, and reduced order models can often be more manageable to work with. The reduced approach is not quite as simple as input and output, but does make some assumptions to simplify structural models. So I've been talking about reduced order modeling, but what actually is it? And I want to preface this discussion just by mentioning that I'll be talking about ROM and mathematics at a fairly introductory level, which may be review or familiar for some of you. So to understand what a reduced order approach looks like, first we need to distinguish them from full order models. So full order models are created by transforming continuous variables, models, or functions into discrete forms using numerical methods such as finite element. As a result, large amounts of time and resources may be required to run these simulations, whereas for full order models, and this can make it difficult to implement them in clinical settings. Reduced order models can help mitigate this by using a smaller number of degrees of freedom, so compared to their finite element counterparts. So the linear systems that must be solved in each iteration are smaller as well. So to put things into perspective, degrees of freedom for infinite elements can be very large in the order of tens to hundreds of thousands, whereas in ROMs, we may only need a few hundred functions to maintain accuracy. And one example of reduced order approach is modeling the effects of blood vessel structure on blood movement. And in this case, a reduced order structural model would reduce the effect of the vessel wall to a thin membrane. Now I've mentioned some of the advantages of ROM already, and here are several more. So other than being less resource intensive, reduced order models have the potential to operate in near real time. And ROMs are also less computationally and demanding than their full order model counterparts. And they can also predict variables such as flow rates and pressure drops with fairly good accuracy. However, these advantages are associated with trade-offs, not just limited to the ones that I have here. So most notably, reduced order models may have larger approximation errors compared to full order models, and some local details may be lost. So a few additional examples of ROMs currently used in cardiovascular modeling include 0D and 1D models. And one issue that arises when dealing with fluid structure interactions, or FSI, is related to the coupling between fluid and solid. The coupling of fluid and solid is usually done, or is commonly done, using a 3D model, but a reduced order model approach can be taken by modeling the vessel as a membrane and incorporating it as a boundary condition for the fluid equations. The result of this is a Navier-Stokes problem on a steady domain with Robin boundary conditions. And you may recall from Dr. Cartier's presentation from Friday, where he mentioned some of the other boundary conditions that can be imposed as well. Now, I will be talking about a very important equation, which Dr. Cartier also discussed, and just breaking it down into parts that might seem a little bit more familiar for those who took introductory physics recently. So the Navier-Stokes equation is very important because it is able to model the flow of fluid through a tube given boundary conditions. And one way of breaking down this equation is viewing it as a very simplified extended form of Newton's second law. So you can see that the terms represent force is as a product of mass and acceleration. So if we break this down even further, we see that there is a term on the left-hand side, dv over dt, which represents velocity change with time. And on the right-hand side, we have internal forces, which are influenced by mu, or viscosity, the pressure gradient, which will be very, very relevant as we see with our blood vessel, where there are different pressures at the arterial and venous ends, and external forces, which can include things such as gravity. And as Dr. Cartier also mentioned, there is also a continuity element of this that I have not included here. The mathematical systems involved in the creation of the whole RAT model were solved using PM3 solvers. And all of the PM3 codes and programs listed are available on the PM3 website, which I believe Tim has just sent into the chat. Also, if you're interested in learning more about partial and ordinary differential equations, I highly recommend visiting PM3's YouTube channel and watching Dr. Karchi's videos on ODEs and PDEs. For data collection, all experimental data were collected by Dr. Barry Jansen, a KCRU researcher and experimentalist. And Dr. Jansen performed the in vivo and in vitro experiments and shared the data with us. So first, surgery was performed on rats to implement a catheter into the carotid artery so that direct measurements could be taken. Clinically relevant measurables were recorded from the onset of the surgical procedure. After catheterization, baseline observations were obtained, and sham procedures were performed on all rats except for 2 that began hemodialysis, or HD, directly after baseline. HD began 1 hour after sham and continued for 2 hours. During HD, body temperature was set at 36.5 degrees Celsius and dialysate was at room temperature. Throughout the HD session, measurements were collected for blood pressure, heart rate, hematocrit, and hemoglobin. And to complement the in vivo experiments, in vitro fluid dynamic experiments were conducted as well, except the animal was replaced with a fluid-filled test tube. And the setup for these in vitro experiments is shown on the right. All experiments use polysulfone-based dialyzers. Prior to data visualization, PM3 software was used to organize the data in preparation for MATLAB figure generation. And figures were generated by reading large experimental data sets into MATLAB. Individual data for each rat were organized so the outputs for each individual rat's measurements could be coordinated and evaluated across variables. And you'll see this in the subsequent figures as the color coding. So these figures represent hemodynamic data obtained from the in vivo experiments previously discussed. A composite rat model was developed that consists of whole body circulation model with body-wide autoregulation and baroreflex control. The model itself is coupled to a virtual dialyzer unit. And the image on the right shows a schematic of the systemic circulation model with arrows representing the direction of blood flow in the arteries, shown in red, and veins in blue. The location of the peripheral dialyzer is also shown. In addition, clinical measurables will be personalized to each rat will be optimized using model algorithms. And these will be used to estimate model parameters. I would like to note that this schematic was adapted from our human circulation model and is just a representation of what we intend for our rat model to look like. Several clinically relevant model outputs were measured, including blood pressure, heart rate, hemoglobin, and hematocrit. Because kidney filtration and circulation are interdependent, hemodynamics are extremely relevant predictors of renal function. And hemodynamics refers to the study of blood flow. And one way of thinking about it is to consider the physical factors that affect blood flow. And these physical factors are analogous to those of an electrical circuit, which is described in physics by Ohm's law. So the electrical current I is equal to the voltage or potential difference divided by the resistance R. And similarly, blood flow, represented by Q, is equal to the pressure gradient, which is the difference between arterial pressure and venous pressure along the length of a given length of a vessel divided by resistance. And although there are other factors that affect blood flow that are not listed here, based on this relationship alone, we can identify a few potential risks for ischemia. And ischemia is a condition where blood flow to one part of the body is reduced. And one of these is low blood pressure. Because hemodialysis involves the removal or the temporary removal of blood from the body, the actual volume of blood in the body will decrease and fluctuate during HD, which will result in a decrease in pressure. And patients receiving HD for end-stage renal disease may also have higher peripheral vascular resistance, which can lead to increased resistance and pulsatility after HD. So based on this equation alone, you can identify that increased pressure reduction in pressure or an increase in resistance can both lead to reduced blood flow. So this reduced blood flow can then cause decreased tissue perfusion and less oxygen being carried to all parts of the body, resulting in ischemia. 4 vascular pressures were recorded over the course of HD, including the pressures shown in panels A, B, C, and D. The oscillations observed for the mean pressure, systolic pressure, and diastolic pressure suggest that a significant amount of individual variation is present. And in contrast, pulse pressure remained fairly constant and with a smaller spread. However, the spikes in blood pressure are followed by immediate drops and don't appear to deviate from the overall trend in pressure. One possible explanation for this is the presence of negative feedback regulation, specifically barrel reflex control. And based on this mechanism, regular oscillation patterns may suggest functional barrel reflex responses, whereas aberrant or irregular patterns may indicate impaired barrel reflex function. The effects of hemodialysis on heart rate are illustrated here. The spread of individual heart rate was highly variable, with some rats showing very wide ranges as seen in the purple and orange, while others had ranges that were more narrow, such as the olive green and dark blue. However, most rats did experience many different modes of oscillation. And overall, aside from these individual differences in oscillation, heart rate remained relatively constant throughout HD sessions. It is also worth noting that these oscillations may represent heart rate variability, or HRV, and the change in HRV before and after HD is considered a useful clinical marker. The results of hemodialysis on hemoglobin and hematocrit are shown here. Unlike the blood pressure and heart rate data, hematocrit and hemoglobin data were collected at discrete times between baseline and 2 hours post-HD. Measurements were taken for the blood entering the dialyzer, which is abbreviated pre, or also known as the inflow, and also data were collected for post, abbreviated post, which is representing the outflow. And generally the same trend was observed between 1 hour pre and post as that of 2 hours pre and post. And as an example of what I mean by this, if we take a look at the pink line shown here where my mouse is, we see that it is increasing from 1 hour pre to 1 hour post. And it is also increasing from 2 hours pre to post. And similar trends were observed for hematocrit as those seen in hemoglobin and in the same direction. And furthermore, the spread of hematocrit and hemoglobin data was wider at the end of hemodialysis at 2 hours post than at baseline. And we can see that as the points at 2 hours post being more spread out than at baseline, where the points are clustered closer together. The development of our whole rat circulation model is currently a work in progress. We have started on the microvasculature components, as Gabrielle discussed in her workshop. And moving forward, our next steps include running simulations, which Tim will take the lead on, and interpreting our simulated data. And ultimately, our goal is to use our 0D rat model to inform future clinical data interpretations from rat to human patient populations. Now that you've heard a bit from us about some of the research we do here at PM3 Lab, I want to give you all the chance to share your feedback. So based on listening to some of the other presentations over the course of the past 3 days, our question to you is, what area of PM3 Lab's work would you like to learn more about? To respond, please visit the link that Tim has sent to the chat, or you can go to www.menti.com and enter the code shown on the screen. You can also scan the QR code if you prefer. And I will navigate to this page to show you what it looks like from your end and how you might be able to submit. So if you navigate to this page, you can see that you can submit multiple answers here, and you're welcome to submit more than one response too. So I will give it 1 or 2 minutes, and then you can also see the results on your end. All right, so just so you can see what the results look like, I will enter something as well so that you can see the display. So I would like to learn more about maybe higher order models, so finite element or finite differences. And then if we would like to take a look at the results. Okay, so FE models, we have parameter estimation, cardiac strain, finite differences, microvasculature, reduced order models, and experimental skills. So it looks like we have quite a diverse responses here, and you can continue entering and submitting your feedback as you like. However, I will proceed with the presentation. So thank you for responding and participating in this poll. So now you may be wondering what the purpose of that activity was. And it wasn't just to create a colorful word cloud. I want to show you where you can find the resources to learn about some of the topics that you mentioned or that you said you were curious about. So PM3 Lab has a YouTube channel, which we've been alluding to over the past 3 days. And this YouTube channel features presentations by the PM3 Lab members and collaborators. And in the area of workshops and tutorials, we have videos on centerline extraction of method and cerebral aneurysm segmentation, as well as digitizing ion channel data of atrial cardiomyocytes using the SCANIT program, automatic segmentation of microvasculature networks. So for those that wanted to learn more about microvasculature, we do have videos on that too, and symbascular modeling of coronary arteries. Aside from the workshops and tutorials, you can find conference presentations such as the Engineering in Medicine and Biology Conference, or the IEEE EMBC 2020, and LHRD 2021, which is the London Health Research Day. There are also many lab talks, including the biophysics of blood flow, which was given by Dr. Dan Goldman, I believe, and you can learn how to engage further with our group. And you can also find lessons on ODEs and PDEs, as well as all of the current 2021 PM3 workshop presentations, which will be uploaded by the end of the day today. And you can also rewatch the previous year's 2020 workshop videos as well. So now, to help you familiarize with where you might find some of these things, I will take you on a tour to our YouTube page directly. So if you click on the link that has been sent in the chat, that Tim has just sent in the chat to our YouTube channel, you can access the homepage. It might take you to the homepage where you can find all of our recent uploads, including, you might recognize some of the presentations just from this weekend. And you can also go to the videos tab where you'll see all of the videos that we have on this channel. And under playlists, for your convenience, you can easily access the 2020 workshop videos, as well as the EMBS or IEEE conference presentations. So I hope that helps with finding where the resources that you are looking for are. And going back to my presentation, I will move on by discussing our achievements so far. So all of these achievements were highly collaborative efforts. And last summer, I assisted with the development of a 0D human circulation model. And the 0D model is discussed further in the paper titled "Using a Human Circulation Mathematical Model to Simulate the Effects of Hemodialysis and Therapeutic Hypothermia." And this was submitted as a preprint recently to bioRxives. And earlier this year, I had the opportunity to present at the London Health Research Day conference in the first ever virtual iteration. The topic of my poster and oral presentation was understanding clinical trial observations using data science. And that brings us to today, where we are currently working on our 0D rat project. And our current project focuses on the prospective modeling of rat hemodynamic and specifically model validation and prediction. And in the future, the current model can be expanded by incorporating other feedback mechanisms such as arterial tone, renal cerebral, while further refining the anticipated autoregulation and baroreflex. This summer, I have also been collaborating with my fellow lab member, Lawrence Liu, who you may have just heard from, and to assist with the development of a 1D aorta cerebral model that can assist with evaluating the effectiveness of treatments for ischemic stroke. In addition, I will extend the established reduced order models to 1D models to obtain even more physiological information. I would like to thank everyone here today, and I also mention that I am very grateful for support from the Kidney Clinical Research Unit led by Director Dr. Chris McIntyre. I want to thank Dr. Chris McIntyre for providing opportunities for me as a trainee and for his continued support and leadership in the field. During these 2 years, I have had the privilege of working with many talented students and researchers who have mentored me and helped me develop professionally as a trainee. Lastly, I would like to thank Dr. Karche for all of his guidance and for providing all of us students with a platform to share our knowledge with the world. Thank you. I will now take any questions.
So just to warm things up, Clara, I'll start by criticizing you, of course. I'll start by criticizing you. On your slide number 16, you sort of provided a brief summary of, I believe, achievements so far.
Yes.
Now, you should bear in mind that, can we see your slide number 16, please? Just to remind everyone. So firstly, I'm glad to report that your first entry at the top is intensely under revision in preparation for publication right now.
Okay.
Tim is going to disappear any minute to get started with it, okay?
Yeah.
And the reason Jeremiah is not here is because he has already hopefully gotten started with it.
Okay.
And so it's no longer a bioarchives. You can have that as a bioarchives preprint. That's okay. That's fine. Okay. But more importantly, there are other critical strengths in your last entry, something that Tim is working on right now. Okay.
Yeah.
Which we must acknowledge. There are several other co-authors that you have. Okay.
Yeah.
One of which a necessary, not highly desirable, but a very necessary strength is our Dr. Goldman.
Okay.
Okay. So it's not that we just like some random authors. There are some critical components that we have to acknowledge. Okay. Without
Yes.
those forces, we could not get here. Fine. So that was just a little bit of a criticism. Okay. And to continue in the same spirit, can we go to slides 10 and 11, please?
Yes.
So we can see the data.
Okay. Let me just,
11.
I will.
10 and 11. So this then goes into I can only see part of your slide, Clara. Okay, and one before. So this argument applies to both these slides, 10 and 11. I'm sure you've said it and that I missed it, but we, as in you, me, and Tim, we have to remember that there are several unexplained gaps in this data that I have forcibly removed to construct these graphs. Okay?
Yes, that's correct. I
That's right.
believe the
So
gaps.
I have forcibly removed those. We have been offered a guiding suggestion by the experimentalist, whoever the experimentalist might be, is that most of these time gaps lasted for round about 1 or 2 minutes, maybe 5 minutes. And those gaps constitute formation of clots, especially in the artificial kidney outside the mouse.
Yes.
Okay, so in all these 4 graphs, as well as on the graph on your next page, there are significant sort of gaps in the actual recording, which I have removed to enable us to make some graph of some description. Okay, I believe Tim is right now refining all of that and putting it all straight. Yeah, but we have to remember that. But importantly, and sort of very significantly, we have to consider this. There is no going around this, Clara and Tim. There is totally no going around this, in that yesterday or day before, Dr. Frisbee told us something really critical. He goes, if you take a rat and hold it down, shake it, slap it a bit, shake its head, twist its head, the stress alone is likely to cause hemodynamic changes, never mind the application of a dialyzer unit.
Yes, that's
The
a
stress alone is going to, so when we are looking for mechanistic cause-effect relationships, we have to phase in the factor that these effects that you've seen may not be because of the external plastic tube that you have.
Yes, that's
okay.
Important consideration.
So I'm not saying this is your responsibility, Clara, but since at least 4 of us are already present here who are involved in this, I thought we should bear this in mind. Okay, because I believe that sort of the stress factor may or may not be actually something that others call ischemia.
Yeah.
And I choose to call things what it is as much as I can help it. Not that I know everything, but let's call it what it is. So this may or may not be an effect of dialysis directly. Just take a rat, pin it down to an experimental board, and the changes in these hemodynamic factors might come out all the same. Okay.
Yeah.
And so those were the two comments that came to my mind. But a final comment has already come to my mind. In that, do you ever stop? Do you ever have some rest, or do you go to sleep or anything or whatever?
Yes, I do.
Yeah, I hope so. I hope so. On one hand, I appreciate your energy, but for you to be putting in so much energy, you also have to pace yourself somewhat. Okay.
Yes.
All
Thank
right.
you,
Well
Dr.
done,
Pritchard.
and I'm thankful for you. I'm thankful to you. Thank you, Clara.
Thank you.
I have a question, I think just kind of jumping off of what Sanjay was saying, is what kind of information then do you think we might be able to determine from some of the control data? Like, do you think from your analysis of the data so far, do you think that how we would be able to apply that?
Yeah, I think that's important to consider, because just doing the analysis and needing to bridge how this analysis that I did will be connected to our simulations. I think that the clinical or in vivo data can be used to just inform those estimates for what kind of parameters or the range of parameters that might be appropriate to use. And I guess the considerations though now are just how much of that is just due to the operation itself, and how much of those differences are just because the rat is exposed to this new environment versus actually due to the hemodialysis procedure. So.
Yeah, that's good. And I think also just kind of looking at the variability that the procedure itself introduces into the outcomes of the experiment, right?
Yeah, for sure.
And seeing if we can, how reliably we can actually say, this dialysis procedure caused this in the hemodynamic response, rather than it could just be like some variability within the data because of the experiment, right?
Definitely. I think there were 11 rats used for this experiment, so definitely a lot of it could be due to individual variability, and I think that's important to consider.
Yeah, and I think that's something that we could definitely quantify and say, well, there's this much variability that we see in the controls versus the experiment.
Yes.
Right.
Tim, could you clarify what you mean by controls?
Well, are there no...
Yes, I have forgotten that Clara's already shown us what control means.
Well, rats that are not undergoing the hemodialysis?
I don't believe we have such data.
Yeah,
oh, no?
Sorry. To clarify, I think I mentioned that there were 2 rats that went straight into the hemodialysis session without the sham procedure, but all rats did in this experiment.
They all have the plastic tube outside.
Right. Okay.
There's no rat that is just like, yeah, okay, give me a measurement here.
Okay.
There's no such rat.
Well, I think there are, we could at least take a look at, similar published data of the same procedure and see just in the literature what kind of variability that introduces.
We are going to have to apply ourselves thoroughly here to look for data in the literature, because I cannot stress enough as regards the novelty of these experiments. Our model may be something that scaffolds this novel experimental thing.
Yeah.
That then makes our model very novel. But we have to be wary of what literature might be able to give us or just cannot give us.
Yeah. I think even just getting sort of an indication though would definitely inform the kind of results that we're drawing.
My most ignorant and straightforward take on how to use the data in your model would be you already have cardiac elastocies, which have a manageable number of parameters there. And you have heart rate defined by Clara's graphs.
Mhm.
So straightforward. And then you have a whole body circulation model, and Clara's gone and made some measurements in some specific location somewhere. And so that could be something that you match up.
Yeah,
I'm strongly opposed to quantitatively estimating parameters just based on these 5 or 10 graphs. That's not a route I'm willing to go down.
yeah.
Maybe Dr. Goldman has something to say about it, but I'm not quantitatively estimating parameters at this stage. We are not there yet.
Okay. So I was just wondering, I mean, are you getting the same amount of sham data that you're getting initially of the dialysis data?
Yes, I believe the sham data is just one of the time points, or sorry, not one of the time points, but all the rats except for 2 of them we did collect sham data from.
Okay. Now, I mean, there's the surgery and then there's the dialysis, right?
Yeah.
Is that right or no?
Yes, that's correct. The surgery, and then in between dialysis was the sham procedure.
Which is, what is that exactly?
I think, I'm sorry, I actually don't know the details of the sham procedure, but in the notes given to us by the experimentalist, Dr. Jensen mentioned the sham procedure before the dialysis session. So I can follow up if I'm actually not sure. Maybe Dr. Karche, do you know what the sham procedure might have been?
So I don't know. It's too early in the day for me to remember this kind of detail.
Okay, but sorry, my point is though, that, I mean, sham isn't just, you can't just do sham and say, I did sham. You basically have to say, you know, I treat this rat this way and I call it sham and I watch what happens. And I treat this way by dialysis and I watch what happens. And then I can compare the two.
Yes, it's usually used as
So I
like a
can
control.
comment on that if you want, Prof, in that we do not on the experimental side, we do not question about what happens when you attach a tube or a procedure. What we're looking to elicit is the difference between 2 synthetic polymers and their function. So essentially what it means for us as modelers in the near future, as in next year or so, is that one polymer has a certain distribution of pores on its surface, and another polymer has a different distribution, as well as the pore shapes and sizes on these 2 polymers are different. And those quantities are going to be compared in relation to the rat model to demonstrate which one is more suitable to use the rat as a recontrollable experimental model of dialysis.
Okay.
So why biomembranes are not used, I do not know. Personally, if I had the kit, I would start with peritoneal dialysis because that's the easiest thing. That's the easiest thing. You just put some liquid inside of the corpus and you let the biomembranes do that trick. But here we are concerned with synthetic polymers, polysulfane and trisulfane or tedisulfane, something like that.
All right, well, if there's no more questions, I think that now is a good time to start our midday break.